Action. So this is Kelly Marcom, for the purposes of this talk, Director of the Hereditary Cancer Clinic. So I'm going to go ahead and start. And I will probably actually be relatively brief because I have to get back to the clinic before people start rebelling. Did not realize this was timed with Go Live Day, or I might have pled for a reprise of Sarah. So any lacking quality of slides or lacking too many slides is the fault of Go Live. We can blame Meister for lots of things. So I think the official title of this talk is Genetic Basis of Cancer. I've always taken that for the purposes of this talk as kind of a discussion about germline cancer syndromes and how that's really enlightened our understanding about cancer biology over the years. So next one. I think there's a smart phrase to advance the slides. There you go. All right. It's not advance. That's exactly right. So we all think of cancer as a genetic disease now, but it wasn't always so. It really took a while for people to come to the conclusion and understanding that that's what cancer is. And so I always like to start this talk off with this pedigree. Any of you have ever seen this before? Has anybody ever seen this slide before? So this is Broca's wife's family, and he took this pedigree back in 1866, and it's historically interesting for two reasons. First, that he did it at that time, even though he's mostly known as a neurologist, of course. But all the black dots were basically women in his wife's family who had had breast cancer. And there are others, the hash-marked ones are people who had other cancers or had liver cancer, probably metastatic disease. So a pretty high probability that this is a BRCA1 and 2 family, but certainly nobody appreciated that at the time. And it's interesting not just that he did this and published it, but then in 1972, Henry Lynch, who's kind of the godfather of cancer genetic predisposition syndromes, famous for Lynch syndrome, et cetera, published this in an article in JAMA. And at that point in time, it's actually the case that a lot of people were still thinking of cancerogenesis as being primarily a viral phenomenon. I looked at the NCI budget and portfolio of activities, and it was heavily biased towards viral etiologies of cancer, not so much thinking about genetics, genetic predisposition. And so he published his pedigree as an example of what the importance of genetics in cancer. So next slide. And around the time, of course, is when Knudsen was doing his work with retinoblastoma and made the observation about inherited retinoblastoma syndrome and formulated his double hit hypothesis for losing a tumor suppressor gene. And so the whole idea of a tumor suppressor gene really stems from the understanding of cancer genetics and inherited cancer genetic predisposition. Next one. And so basically the idea that and most people have two normal copies, and you have to lose one copy of the tumor suppressor gene to begin on the pathway and lose a second to then go on and develop a cancer. Whereas in familial syndromes, all the tissues start off heterozygous for one mutant allele and then lose another, and so we're all primed in certain tissues. It's obviously more complicated than this, but primed in certain tissues to develop cancer. Next. Go. That's really led into the whole Vogelgram of progressive DNA damage. Next, some animation on this slide, and of course I apologize, of developing additional phenotypic features as a consequence of accumulation of genetic damage. Next. And now we know that that's not just straight up DNA base pair damage, loss of DNA, but lots of epigenetic changes as well that all feed into immunomodulation and everything. But it stems from the germline cancer genetics observation. So it's important to know some about these syndromes, even if you might think that clinically they're not common every day that you see them. In point of fact, probably actually do see them every day, and they need to learn to be able to recognize them. Next. Next. Okay. So the next heyday then historically really came about in the 1990s after all the genomic revolution happened and we were beginning to sequence the human genome and the ability to do linkage mapping in high-risk families. So this is the cover of Science that had the first publication of BRCA1. The lower one, first author is Andy Cottrell, who was actually at NIEHS when he did that. He came here then and actually was here when he was one of the main people who cloned BRCA2. So Duke actually contributed a lot of the families to this, to the initial linkage studies and was part of that consortium. Next. So that all happened, through the 90s then. All these autosomal dominant cancer predisposing syndromes were identified. And the next two slides really are the ones that are probably most important to know. So BA1 and 2, of course, I think you all are familiar with, primarily breast and ovary, Li-Fraumeni syndrome, mutations in P53, primarily with sarcomas, breast cancer, leukemia, lymphoma, and adrenocortical tumors. CHEK2, which is a Li-Fraumeni-like syndrome, Cowden syndrome, which mutations in the PTEN gene. P-Jaeger primarily predisposes to GI malignancy, but also breast cancer and pancreatic cancer with mutations in the STK11 gene. Next. And of course, the hereditary non-polyposis cancer syndrome with mutations in all the various members of the mismatch repair DNA pathway. Endocrine syndromes are important as well. So MEN1, mutations in the menin gene or CDKN1B, and then MEN2 with mutations in the RET gene. Some overlap in endocrine sites like overlap parathyroid tumors, thyroid cancers, but slightly different syndromes otherwise as far as the sites that are affected. But if you look through this list, it's important to note there's just an awful lot of overlap in tissue sites. And why given genes and germline mutations necessarily have a higher preponderance of mutations or cancer events in certain tissues, it's really still being sorted out. But none of these are kind of pure syndromes that really affect only one organ system. If you've got a mutation in one of these inherited cancer genes, it's many different tissue sites that can be affected. Next. So I always defend because the person at Duke who kind of led the consortium effort was Dirk Eigelhart, who was the breast surgeon at Duke and then went up to Harvard. So I always liked this because it was Dirk bringing his family tree to class, and he was probably the person that got me most involved in setting up the hereditary cancer clinic and working on this. And I always have to make a nod in this. Clinically, I think increasingly everybody understands the importance of family history. Now we have this great new tool in EPIC. So if you have family history, okay, maybe it's a marginal tool in EPIC. But it is important, and particularly in the oncology clinics, to make note. And it doesn't take that long to take a 3-generation history. The thing, and to get some idea of the denominator of family members as well. So oftentimes, people just put, no family history cancer, no in our clinic, no breast or ovarian cancer. But it's one thing if I'm sitting there looking at a 35-year-old woman who's got only brothers or, only child, mother, and father, versus someone who's got, 12 sisters and 20 aunts on each side. So the denominator is important when you try to put things in quantitative models. And I know it may seem like it's not the most important issue when you're working up a new patient, but really, now they're just going to have to put Marcus reviewed for everybody's past medical surgery. It takes time to get the detail on the family history. You want to know whether there are any bilateral cancers in the family, ages of diagnosis, and particularly unusual cancers that would map onto any of those other syndromes. And sometimes those unusual cancers are not necessarily what you would think, like invasive lobular cancer. I've got one family, it's 3 generations of invasive lobular cancer that we're still working out now. But that's associated with E-cadherin mutations as a particular phenotype or particular histologic type for breast cancer. So asking those kinds of questions is important. Now, obviously, not all families are going to know them. Most families aren't going to know them, but at least they can look into it, and sometimes they can find out more than you think they're going to be able to. Next. All right. So, sorry, I jump around a little bit in this when I was putting these slides together last night, but every year, we update. The question always comes up when you're talking about these syndromes. Well, who should you send? Who should you send genetic testing on? We make an argument in our academic center. It's not really who would you send genetic testing on. It's really who should you send for genetic counseling so that they can really do the more detailed detective work and getting the family history put together and running the probability models and really deciding which syndromes to go after first. That's the current state of affairs, but as I'll talk about at the end of the talk, that's pretty rapidly changing. So invest in ovarian cancer in the breast clinic. Obviously we look for early age of onset, but now increasingly we also look for anybody with triple-negative breast cancer. So we, as a general policy, anybody under the age of 60 with triple-negative breast cancer is worth consulting with the genetic counselors. And with those patients, we do go on to test as well. We basically test all ovarian cancers. We test almost all male breast cancer. And then beyond that, it's looking for any of the constellations of those other cancers in whatever it is, 4.6, with a combination of other malignancies that are associated with any of the syndromes that we talked about. You always test the affected individual in the family as well. You never ignore the paternal history. Those are common misconceptions I think probably most of you are aware of. But if you have somebody who's unaffected in the family, you really are left with an uninformative test, because if you don't find a mutation, all these are really Mendelian disorders, you just may have missed the person, the unaffected person may just not have gotten the gene in the family. So it's always much more helpful to test the affected individual to get a truly informative test. Next. This just elaborates then once someone's been seen, what we actually look at for testing criteria beyond that. So, as I mentioned, for example, under the age of 60 with triple-negative breast cancer, almost regardless of family history, if we can get it covered as an indication for testing, any combination of breast and ovarian and close relatives, those under the age of 50 with 2 members in the family. Almost all those criteria, if you meet them, will get someone covered for being able to do, you get insurance coverage for BRCA1 and 2 testing. So sometimes you come on a family and it's obvious, although sometimes you still come on families like this, and despite the fact that any of you taking that family history in clinic would hopefully say, breast cancer at age 32, mother 29 and 32, aunt at age 50, one unaffected aunt, and then you start going back to unknown primaries and the grandparents and great-grandparents. I mean, that's pretty obvious, and this family did have a deletional mutation in BRCA1, clearly deleterious mutation. Next. So those we're trying not to miss now. But then, it's things like this now, we're trying to key off of histologic features like triple-negative disease, like invasive lobular cancer, certain types of thyroid cancer, where it's a little less obvious, but these are just our numbers from the HCC clinic over the past 2 years. So if you look at our total number of positive breast cancer mutational detection in patients with triple-negative disease, before 2011, when we weren't necessarily testing triple-negative disease quite as much, it was about 25% hit rate for finding a BRCA1, probably a few BRCA2s in there as well, mutation. So that's a pretty high rate. And now that we've expanded those criteria some, it's still about 11%. And we've always taken the stand, if we're hitting an 11% detection rate, we probably still may be testing too few individuals, because that's still a pretty substantial hit rate. And if you look within all breast cancers and start asking this question, how does germline mutational status relate to the type of breast cancer? That Venn diagram kind of gives a sense of it. Within all breast cancers, there's triple-negative breast cancer. Within triple-negative breast cancer, some majority of those are basal-like, and of those basal-likes, some subset are BRCA1 related. So I think that taxonomy of genetic related events and how it relates to subtype is increasingly being better defined. There are some BRCA1 tumors, however, that end up being ER positive or non-triple negative or non-basal-like, but it's probably only about 15 to 20% of the cases that happen in association with BRCA1. The clinical utility of doing germline testing in the single gene autosomal dominant disorder syndromic era is mapped out here. And I think it's safe to say back in the 90s when these genes were first identified, there's a lot of clinical nihilism about doing genetic testing and a lot of statements to the effect of, well, we don't know what to do with this information, why would someone really want to know? But in concerns over discrimination, concerns over patient anxiety and depression from it, from getting a testing result. And over the next 15 years, really, the data came together to show that, first of all, patients, if they know they're at risk, pretty good studies, what was shown is that patients actually have more anxiety and depression if they know they're at risk for carrying a mutation, either because of a known familial mutation or a high probability, and then opt not to have testing. They actually have more trouble with anxiety and depression than individuals who get tested and they're shown to have a mutation. As well, issues about discrimination really not come to pass, certainly from the standpoint of rushing me along, Jim. Maybe appropriately so. But from a health insurance standpoint, we haven't. I think we have one patient, probably 10 years ago, who came back to us and said they'd had some issue with insurance coverage on the basis of having been tested or having found to have a mutation. And I think there are a variety of reasons for it, but editorials written 5-6 years ago, that made the point that who would be stupid enough to let it be known that they were discriminating on the basis of BRCA1 and 2 gene mutation testing? Can you imagine what kind of mob you would have marching on your insurance headquarters if you got caught out doing that? I mean, I really think that enters into it, but the more calculating is that I think if you run the actuarial numbers, it's worth their efforts to identify people who are predisposed. They'd much rather pay for their prophylactic surgery and even their screening MRIs than to pay for their chemotherapy and endocrine therapy and surgery. So we haven't really seen that be an issue. And then including we had data about screening, including MRI screening and to some degree tamoxifen as a prevention agent. And at the same time, increasingly a better understanding about the mechanism by which mutations in BRCA1 and 2 predispose to breast cancer. In recent years, then, of course, that's translated into the development of the PARP inhibitors. So 2 years ago, I think this was in some ways a hotter topic. The PARP inhibitors in breast cancer got a little bit of a black eye because we had one trial, the drug that showed up nameless, that was touted to be a PARP inhibitor, but wasn't really a PARP inhibitor when it came down to it. And so I think a lot of people walked away from that experience thinking that PARP inhibition maybe wasn't all that tractable a target. But in the simple, quick and dirty version of synthetic lethality, the importance of the concept here, of course, is that you have pathways that help in repairing double-strand DNA break, one by excision repair and one by homologous recombination. And the dominant one is the homologous recombination pathway, and it's facilitated by both BRCA1 and 2. In the absence of that, if you have a double-strand break, you use more of this base excision repair pathway that's dependent on PARP1. So if you are missing BRCA1 all the way over in the right panel and then inhibit PARP1, and then cause double-strand DNA break repairs in cancer cells, then that leads to apoptotic events and cell death. And that's still a viable theory. And within the context of truly BRCA1 and 2 related cancers, the trials are going forward with primarily with olaparib and veliparib, although there are a couple others in development as well, to test this out. So we have a trial now with metastatic BRCA1 or 2 related breast cancer, treating with veliparib in combination with carbotaxol versus placebo versus temozolomide with veliparib. It's challenging to recruit to, as you might imagine, but probably have somebody we're consenting today for it. So the point is that we went from this kind of total nihilism about knowing this information to now having very specific proactive procedures, screening procedures, and potentially use it as a predictive marker for response to treatment. And so it really shows that BRCA1 and 2 demonstrate a pathway of how understanding that genetic basis can help to develop therapy. Next question. All right, I think I've covered these. Go. So just to kind of the estimates then for breast cancer as to which are BRCA1 and 2 related. If you look overall, probably about 30% of breast cancer is hereditary or familial. Of that 30%, half is BRCA1 or 2 related. So that's somewhere around 5 to 10% of the total. But that leaves a whole swath of other low and high penetrance genes and other cancer syndromes that tie into breast cancer predisposition. Next. And skip. And so I'm going to skip through hereditary colorectal cancer fairly quickly. But, this shows kind of the same distribution for hereditary colorectal syndrome, in which 10 to 30% are familial cases, 2 to 3% that are HNPCC, and 1% that are LFT. So, there's an awful lot of sporadic colon cancer and an awful lot of familial that's still being defined. Next. So, it's important, as I said before, this is another DNA repair pathway with various mutational events in it, where it's multiple sites that can be affected. So not just colon, but endometrium, ovary, stomach, and the GU tract, as well as skin tumor and brain tumors as well. Multiple sites that can be affected as a consequence of having an inherited mutation in one of these DNA mismatch repair genes. Next. Keep going. And this just shows the relative probabilities of lifetime risk any one of those organ sites with mismatch repair mutation. And it varies a little bit depending on which particular gene is mutated. Next. So this is a family on that we identified. So I always like to actually put this pedigree in this talk because the index case here, the 60-year-old woman with uterine colon, is actually on service with lung cancer is a third primary, and it had a daughter who had colon cancer at 35, and then the sister with breast cancer, but then the colon cancers you see in the generation before her. And what's notable is that she was serviced for lung cancer, and yet with this family history, and this is probably 5 years ago now, she had never had testing for mismatch repair mutations, even with that family history. So colon cancer and colon cancer predisposition syndromes, I think, are probably still not identified as currently just basis of routine referrals as are BRCA1 and 2. Next slide. So this just shows kind of the recommendations for follow-up management. Next. If you look then at clinical criteria, there are two main sets of clinical criteria for identifying HNPCC patients: the Amsterdam criteria and the Bethesda criteria. That have those are the criteria in Amsterdam: 3 relatives with some Lynch cancer and 2 generations and 1 diagnosis under 50. So that's a pretty easy set of criteria to wrap in your head. The Bethesda criteria, the revised, just have some particular tumor histology descriptors within the tumor histology, like signet cell features within colon cancer that tie into that as well. But because the referral patterns for HNPCC testing have always been a little bit more challenging, not as sensitive, a lot of institutions, including ours, we several years ago embarked on a protocol of doing routine testing for all colon cancer cases with mismatch repair mutation by microsatellite instability in immunohistochemistry staining for the mismatch repair proteins. And so we do that now in screening anybody as a newly diagnosed colon cancer patient. Next. So there is some overlap with sporadic tumors that can have microsatellite instability as well. The majority of Lynch-related tumors will have MSI at multiple loci. So generally, you look at a panel of 5 microsatellite markers in order to screen somebody as being MSI positive. And if over 30% of those are unstable, then that's MSI high. And those definitely get screened for germline mutations by protein staining next, and then gene sequencing for whatever protein is missing from the tumor. There may still be some predictive function for mismatch repair-related genes as not being as responsive to 5-FU. So again, an argument for these germline factors is having some predictive function for response to specific therapies. Next. This just shows then the process for immunohistochemistry to be able to evaluate if somebody has an MSI high tumor. Those are the proteins that we screen for initially, MLH1, MSH2, MSH6, and PMS2, and that helps then guide detection of the gene to sequence for mutational sequencing. Most on colon cancer samples now, but there's data now for doing it for many tissue sites, including endometrial samples. So we'll start with the colon cancer in a family if we possibly can. Next. Right. And then just a brief nod to rarer syndromes, Peutz-Jeghers syndrome. I've only seen one patient with this. This is the classic lip pits or pigmented spots on the lips. It's associated with breast, pancreatic, uterine, and lung cancer, as well as colon cancer risk that's the most markedly elevated. Next. And then juvenile polyposis, all falling in kind of this polyposis family of disorders that also increase the risk of colon, but also gastric, small bowel, and pancreatic cancer. Next. Things are rapidly changing though, and we've gone from this era of single Mendelian disorders trying to look for single gene mutational events in syndromic families where there's a clear pattern of cancer, to then, through the 2000s, the approach of using genome-wide association studies to look at loci that were linked to breast cancer risks in high-risk families. And the results of the GWAS studies are now really coming through. So just from this past April, there were two large consortia that were put together for basically getting all the groups in the world that had their high-risk families and had DNA and had phenotype information, and doing these genome-wide association studies to look for new loci that are now beginning to percolate into clinical use. So it's really been over the last 2-3 years that these studies have started to come through. This one, published in April of this year, identified 42 new breast risk loci. Now, none of these have the same kind of odds ratios for developing breast cancer over the course of someone's lifetime, like BRCA1 of 70 to 80%, and BRCA2 of 50%. These are all odds ratios that are more in the 1.3, 1.4 range, so 20 to 30% increased risk of developing breast cancer. But we were able to find some that were associated with specific phenotypes like ER-negative disease. And there was another group that Andy Burchuk here and Joellen Schildkraut were really lead investigators on that was able to find similar loci for ovarian cancer risk. Now, next slide. The question is, though, what's the approach for being able to screen for these things? Now, so of syndromes I've talked about, this slide actually is a bit of an old review now, but it makes the point that if you think about most of these familial syndromes, they're associated with mutational events that are in DNA repair pathways, either double-strand break or mismatch repair. So BRCA1 and 2 and the HMPCC genes are kind of there in the center, but all the other genes that are associated with some degree of increased risk really are genes that predominantly interact with them. So, for example, P53 interacts with BRCA1 in monitoring for DNA damage, and clearly, it's an inherited predisposition gene as well. The ATM family members in both heterozygotes and homozygotes, tie into the same type of DNA repair. All these, many of these genes also are, of course, in the Fanconi's anemia family of disorders, that if you have mutational events in two genes, you can have Fanconi's anemia in childhood. So the general theme for cancer predispositions, the majority of these syndromes that are the strong autosomal dominant ones are related to DNA repair mutations. There are some exceptions, like ecadherin, predisposing to gastric and lobular cancer. It's not. It's more of a cell signaling gene. You know, the RET gene is more of a membrane signaling gene as well, but ones that I think are the greatest clinical importance are all DNA repair pathways. Next. Caldome I never know where to put in. So it's relevant because it's in mutations in the PTEN gene. It's an increased risk of breast cancer and thyroid cancer. So again, an example of a disorder that has both these malignant predispositions as well as these benign skin features and hamartomatous polyps that are part of the syndrome. And why those particular tissues and not others are predominantly represented for the cancer risk is hard to say, but important just because it's an inherited mutation in the PI3 kinase PTEN in pathway. It is relatively rare, and we've probably tested 100 people for Cowden syndrome now, including one patient of mine who's had bilateral breast cancer and thyroid cancer and has these characteristic skin tricholamomas, which are these skin tags in the face. And in the best research lab and clinical lab, they couldn't find where her PTEN mutation was. So I know the syndrome exists, but it is very hard to find individuals who have it, even when they seemingly have the full phenotype. Next slide. Next. Leishmania already talked about a bit. Next. Put this in there, yeah. I won't tell you this story. It's a long one. So where are we going? It's a very interesting one, but it's a very long one. So what's changing all this now, of course, is if you think about everything I've shown you, it's based on taking in a family history, looking at patterns of cancers, and then trying to figure out what genes you ought to test somebody for. But now, with the collapse in cost for doing full gene sequencing, the ability to, rather than just trying to one-off each one of these mutational events, potential mutations, sending people through sequentially and really going through what can be a somewhat frustrating process of doing one gene at a time. Very quickly now we're moving into the full genome sequencing era because the costs just are allowing to do it. Has everybody seen this slide? It's shown a lot now, but it's comparing this to Moore's Law, the cost of computer chip storage. And Moore's Law was this exponential collapse in the cost of computer memory. Well, the green line that charts the cost for getting sequencing information for a whole genome goes from $100 million to now close to $1,000 in a 10-year time period. So it blew away more comparison to Moore's Law, even though that was pretty impressive itself. And next. So this is just another way of showing all those different members of the DNA pathway and kind of the relative risk that you get with mutational events in them for developing breast cancer in this case, just to make that point a bit more. Next. So where we're going really is the first step is a lot of companies now are offering panels of testing instead. So this is one company, Ambry Genetics, that we work with that will sequence all of these genes to mutational information. Look for those things that are more moderate penetrance mutational events that may inform cancers that have multiple cancers in them. So this is their breast panel. They have a cancer next panel they do that has a bit more of the mismatch repair mutations in most of these genes on it as well. So there's still a little bit of picking depending on what type of pattern you're seeing in the family. But this is the next step into going on to doing full gene sequencing. You'll notice, of course, what's missing from this slide.
Will they add it though now?
Yes. In fact, I kid you not, it was 30 minutes after the Supreme Court decision came down. They'd sent out an email that they now are offering BRCA 1 and 2 testing on their panel. And in fact, they actually always had the sequence information. And apparently they had on occasions told people BRCA 1 and 2 results even before the Supreme Court decision. But yeah, I mean, the Supreme Court decision we could talk about for a while, where that, how that's impacted, how the patent impacted the implementation of BRCA 1 and 2 testing, contrasting that with what's going on for hereditary colon cancer, where the patent was held by Johns Hopkins and Vogelstein's group, and they never enforced it. So everybody could offer testing for the mismatch repair genes. But it was interesting because, BRCA 1 and 2 testing kind of took off, And colon cancer testing never did. And I have to give Myriad some credit for at least having put the testing out there. Now, whether they did it responsibly or not, we could talk about as well. But regardless, they did at least get it out there, so people were being tested and screened, and I think to good effect. But now, the competitive environment's going to change drastically, and clearly the implications for that on the cost of testing should be beneficial to patients. So right now, BRCA1 and 2 testing from Myriad, doing testing alone is, about $4,000. And depending on someone's insurance, they pay, very little of that, or they don't get it covered at all. These panels are probably going to come in at about the same price point to begin with, but I expect that that's probably going to go down pretty quickly based on the cost of sequencing. And Myriad also is now, in fact, we've had discussions with them. They have their beta panel of genes that they haven't fully released what all they're going to test yet, but starting this fall, they'll be doing panel testing too. And I imagine everybody will pretty quickly move into doing more full genome testing, but of course that opens up a whole other realm of uncharacterized variants and results that are of unclear clinical significance. Next. So that's where we are. That's where we're moving. And to the extent that those things are helpful for screening high-risk patients and understanding the etiology of cancer, particularly in family histories where it's not quite clear what's going on, increasingly, that data set will just have to accrete over time so that we can interpret those results. And I guess based on both the 5FU mismatch repair experience and the PARP inhibitor BRCA1 and 2 experience, I do think those things are going to translate into a predictive understanding of responses to given therapies, both in combination of looking at germline genetic information, but of course coupling it with somatic mutational information as well. And if you look at that full picture, then you begin to get a better understanding of someone's cancer, heterogeneity issues acknowledged, that I think will really enhance treatment options, both for invasive disease and for prevention as well. And I think they're probably pretty close to so I guess the slides that will go around. These are two kind of helpful links. The one in the lower right probably is, as long as it stays funded, if you ever want to know anything about any kind of germline testing, even outside the cancer space, that is a pretty good website. I don't know if any of you know that one. Others are a little bit older now, the Cancer Genetics Web, and OMIM is kind of the historical one that Victor McCusick started back in the 50s. It's just kind of an encyclopedic concatenation of information as it comes in about genes and events, and it gets a little bit unwieldy at times, although it gives you certainly the historical chronology of what was figured out about any given gene and its activity. So that's okay, you're fine, go. All right, so I've pretty much covered that. So I was supposed to make questions. Does everybody else make questions? For big questions, I'm sorry. Okay, if you want to implement a smart phrase, what do you do? I've been a little epic focused of late. So I don't know that this is, the germline stuff is usually not too represented on the boards, but you need to know about mismatch repair and Lynch syndrome. You need to know about BRCA1 and 2, and some about multiple endocrine neoplasia 1 and 2. I know I just very superficially went over it, but those things generally are represented to some degree. So those. Beyond that, they don't get into much depth about the rest. So, any questions? I know I went fast, but I can feel them over there asking somewhere. How do I order? All right. Thank you.